Hello, welcome to the ESDR kitchen. We were just waiting a couple of minutes to get participants being registered. So we are here for the new episode of the recipe book. So in the meaning of the recipe book, we are going into different recipes. So now today it's the turn of Florian Grüber, and I will pass the word to my colleague Enikö Sonkoly from Sweden, and she will introduce him.
Thank you very much, and welcome to this episode of the ESDR Kitchen. It is my great pleasure to introduce to you our speaker today, who is Florian Grüber. Florian Grüber is an associate professor at the Department of Dermatology at the Medical University of Vienna. And he's also head of the Christian Doppler Laboratory for Skin Multimodal Imaging of Aging and Senescence Skin Margin. His research focuses on the interaction of the skin with the environment and also the role that the lipids and the oxidation play in skin biology. So in this episode of the kitchen, I hope that he will reveal the secrets of lipids in the skin, how we can study lipidomics, and so we are very much looking forward to hear the recipe. Very welcome, Florian Grüber. And just one note that for the audience, please ask your questions in the Q&A or in the chat. Florian Grüber, the floor is yours.
Thank you very much, Enikö. Thank you very much, Anna. Let me share my screen. And I want to thank a lot the ESDR, my hosts, and also the sponsors today to allow me to talk today a little bit about lipidomic analysis in skin research and how we applied it for the so-called multimodal imaging of stress-related adaptations to lipids and metabolism in the epidermis. This is not only the 50th episode, as I heard, of this very nice program, and it coincides also with my 50th birthday. So to spare you my aging looks and to give some more bandwidth to this presentation, I will now shortly switch off my video and start with my presentation. The maintenance of lipid homeostasis in our skin, especially within the epidermis, is a complex task because there is this directional differentiation of keratinocytes, and therefore the synthesis and the metabolism of lipids must be fully coordinated between the controlled cell death, and also it has to be coordinated with the tasks of the skin appendages, which provide the surface lipids. During this terminal differentiation of the keratinocytes, there is drastic changes in the lipid composition, and in this granular layer, the intracellular organelles, the laminar bodies are formed, in which the glucosylceramides, phospholipids, and sphingomyelin are stored temporarily. And these need to be metabolized to produce the final lipids of the stratum corneum. And for that, these precursors, for that, these precursors have to be exocytosed together with the enzymes that are needed to metabolize them. So this yields a mixture of free fatty acids and cholesterol and ceramides, which is the say epidermal derived part of the stratum corneum. But these keratinocyte derived lipids, they blend then with the part that comes from the sebaceous glands, which is a mixture that is stronger in triglycerides, wax esters, and squalene. Of course, the main focus has been laid on the changes that the skin and the epidermal lipid composition undergo during the disease of the skin, or the major inflammatory disease of the skin, which again are often correlated with disturbed differentiation and barrier formation. And for example, major shifts in the composition of the ceramides, and of course also in their ordering, they are correlated to the lesional localization and also to the severity of this disease. However, the changes in the lipids are also likely involved in inflammatory events that characterize this disease, both in psoriasis and atopic dermatitis. Lipids that are immunoregulatory eicosanoids, like these hydroxy, the HODs and HEATs, or at least lipids that are structurally similar to them, are often found dysregulated. And most of this, the knowledge on these lipids changes came from sampling from the surface, or from biopsies, or from plasma.
I'm sorry to interrupt.
Yes.
So would you be able to turn on your video if it's possible?
Yeah.
Thank you very much.
I can do that. I just turned it on for the so that we have no problems with the transmission. Okay.
That's fine.
Because yeah. Okay, but the changes in the lipid composition are also beyond the big diseases are interesting and relevant, and especially for us, there is an interest in the extrinsic stressors in diet or aging on lipids that manifests also in biological consequences. And here, the changes in the lipid composition and quality, they are not necessarily the product of, for example, enzymes, as it is in some of the inflammatory disease, but or synthetic or metabolic pathways, but also of chemical processes like oxidation or nitration, chlorination, sulfonation. And the sum of these lipid modifications is called under is named epilipidome. And to study the nature and the action spectrum of the epilipidome, which can reach from roles as for example, as second messengers or to toxic substances, has only become possible through modern, say, analytical techniques. So for most analytical approaches, there has to be some kind of a sampling and isolation and extraction steps in organic solvents. The analytical methods that are used, they strongly differ in the complexity and the demands on the infrastructure, on training, and of course they differ in the type of results they can deliver. So most analyses are done on lipids that are extracted from skins or from skin cells. And there, a great challenge is always to acknowledge that the skin is a heterogeneous organ and tissue, and that there are, say, differentiation-dependent dynamics in the sample that you will be able to take from this organ. And this has to be remembered when performing the analysis. Newer evolving technologies, we will come to that, mass spec imaging, for example, they allow to analyze the lipids in their tissue context. And this new development discloses a big gap because specific lipids, or lipids in general, can rarely be localized and detected with antibodies or other technologies that are useful in histology. And most of the lipidomic methods generate, however, of the new methods generate huge data sets. And so there has to be also an evolution in the say data sets and also in the software that handles these big fat data, and this is thankfully happening. If we look a little bit on the sampling aspect, so first outermost source, the collection of sebum is very easy and can be done non-invasively with adhesive tapes or patches, absorbing patches. Sebum lipids can be washed from hairy body sites of men and mice. Stratum corneum samples, however, are usually obtained then by horizontal sequential sectioning and using adhesive tapes or super glue, so cyanoacrylate, or simply also by physical scraping, for example, with a scalpel. So sebum-derived surface lipids can penetrate into the stratum corneum, and this has to be remembered during the analysis. So the stratum corneum can be obtained also by trypsinization from punch biopsies or from ex vivo explants. And to gain a full epidermal lipid spectrum, what also can be done is to separate the epidermis from the dermis in vivo using suction blister technique. And the good thing about that is that you can also collect interstitial fluid lipids that can also be analyzed. You can also use ex vivo heat separation or the enzyme dispase. For the corneocyte lipid envelope, which is usually these ultra-long ceramides and fatty acids, which are covalently bound to the proteins of the corneocyte envelope, they can from that, or they have to be released by alkaline hydrolysis and can then be extracted with an organic solvent. And these organic solvents are like most samples, also including cultured cells. These extraction protocols are derived from very simple and very old classical methods, which are based on, say, chloroform methanol extraction, for example, the Bly and Dyer method and or the Folsch method. After this initial extraction, one can apply a kind of chromatographic separation or liquid-liquid extraction methods to enrich specific lipid classes. For example, so we have done that in our laboratory, lipid extraction with hexane and acidic methanol can effectively enrich polar lipids, for example, phospholipids from cultured cells of all classes. If we come to the detection methods, of course, the most basic and inexpensive one is a thin layer chromatography. And it's good for fast profiling of lipids in a sample. For example, here you see a ceramide experiment. And this technique also pioneered the research on skin barrier lipids, on metabolism and in pathological changes to the epidermis. In TLC, so thin layer chromatography, you have a mobile phase, a solvent mixture where the lipids are soluted in, which passes over a thin layer of a stationary phase. This is usually a silica gel, and for high performance, this is a very size exact silica. And with this method, you can separate a lipid mixture into its components. And by adapting the mobile phase, you can determine which spectrum of the lipids, for example, from the polarity, is separated best by this method. These separated lipids, as you see them, then here are usually visualized. You burn the whole thing, so it's called a destructive derivatization using an acidic copper sulfate solution, and you then char it in heat, and then you get this coloring. And however, to identify or tentatively identify lipid species with that method, you always need to run also standards in parallel. So here you see ceramide standards and stratum corneum extract, and then using these standards you can do a quick overview, a very rough quantification, but you can only get information about lipid class or maximally the subclass of the lipids there. in contrast in complexity, then is of course mass spectrometry, and mass spectrometry lipidomics allow for detection of the molecule mass to charge ratio for a very large number of lipids. The main prerequisite for that is so you can only measure molecules that can be ionized with the ion source. If you then add in front of that, here we have liquid chromatography. If you couple that to the mass spec, then and to separate the sample before the ionization, then you have a very, very powerful tool to address the complexity, and it has been used frequently to investigate, say, complex composition of the skin lipidome. This ionization that you need to generate the ions that can be detected usually happens so-called electrospray, and then for so-called MSMS, the mass charge ratio, also of the fragments that happen then through collision or after the initial quantification, and this is also recorded by detectors and gives then augments the information, the structural information on the molecules that you investigate. The LC-MS data can be acquired in so-called untargeted or targeted mode. So targeted means that you already kind of know what you're looking for. So targeted analysis is suitable for high throughput quantification of selected lipids in large sample cohorts, and you don't need that much data processing because you already kind of selected the slots. But you must know before, you have to have some knowledge about the structure, about the elemental composition, the MZ, and the expected fragmentation patterns of these molecules. On the other hand, untargeted lipidomics gives you a more kind of general, holistic view of the whole lipidome, but this requires intensive processing of the data, and it also requires that the data are collected at a very high mass resolution and very high accuracy, which can be done by, for example, Orbitrap or QTOF instrumentation. Another approach is that if you omit the liquid chromatography step here and do the separation within the ion source, there is also technologies that can do that. It's called shotgun lipidomics because this liquid chromatography step for each sample is quite time consuming, and this shotgun chromatography is much faster and can give you a large overview about the lipid classes and species, and is very useful for high throughput analysis. But of course, high throughput, these lipidomic experiments, they especially the untargeted ones, result in several thousands of these MSMS spectra, and this is why you need some bioinformatic tools that can handle such amount of data and that can help you filter out relevant information. Over the last years, there have been some tools developed which allow, for example, matching the experimental MSMS data that you generated with something that is stored in libraries, like in lipid maps or in structured databases. There is also software that uses, for example, curated fragmentation rules. So you know in which parts some types of lipids or some types of fatty acids that are attached to the lipids should break. And with these, you can then do with the software that can identify those rules. You can do a bottom-up identification of the ions and the fragmentation signals you have acquired with MSMS. So there one software that can do that is, for example, this Lipid Hunter, or software that combines both of these capabilities, like Lipostar. Of high importance, however, for all these applications is that from starting with the sample collection to storage to analysis, you have to observe clear quality standards that are recorded and also reported, ideally then for publication. And such a minimal reporting standard is promoted by an association of experts that are together in the Lipid Standards Initiative. And so they have some kind of a white paper. So what are the minimal rules that you should observe when you report lipidomic data and claim that what you have detected or quantified is one specific lipid? Of course, these data sets for the lipid molecular species can then be used for quantitative analysis and systems biology. And lipidomics are usually considered to be a subset of metabolomics. And after a clear identification, you can connect them in pathway models and some molecular networks, or connect them also to other omics data that are generated from the very same sample. To get an idea about this myriad of interactions underlying the biological processes, there is some pathway analysis tools that have been developed. And one of these open source and open access pathway tools, which also has a dedicated portal for lipid researchers, is Wikipathways lipid portal. And so one of the typical signaling pathways, for example, which can depict lipid synthesis, receptor binding events, or formation of protein complexes, or phosphorylation, translocation, regulation of downstream genes. And you can depict it in Wikipathways with a minimal set of symbols, lines, and arrows. So while the graphics are not totally flashy, it's a very useful tool, and I just picked randomly something out. I showed in one of the first slides that 7-hydroxycholesterol is dysregulated in psoriasis. And so I fed that in. So taking this example, it was elevated in the plasma of psoriasis patients, and you can then put this molecule into the context of the signaling pathways that are described and deposited here. And one could, for example, investigate other known, say, gene regulations or pathway regulations that are regulated in psoriasis. And in that case, for example, you could then follow up this enzyme here, which degrades or metabolizes this hydroxycholesterol. Is that regulated in psoriasis? And just for fun, I checked it, and indeed, this is decreased in psoriasis. And here is, for example, how you could then starting with the lipidomics data, some investigation of say the signaling and the networks where that is happening. But what we of course want to do is not only signaling, but we want to know where these lipids localize in the skin, what happens there. And to do that, for that purpose, there's an exciting methodology being developed and is evolving. And this is mass spectrometry in high spatial resolution, not to be mixed up with mass cytometry with antibodies and labels, but the mass MS-based imaging, MSI, it performs spot per spot mass specs over the sample area in a matrix, and allows for absolutely label-free and antibody-free detection, identification, and to some extent also quantification of single lipid species. And so a tissue section is generated and then mounted on a slide. And in case of matrix-assisted laser desorption ionization, which is in short MALDI, then this is covered with a so-called matrix. And this matrix can be used to target the ionization characteristics of the sample and of the molecules in the sample. And thereby you can somehow adjust the type of molecules which will be detectable there. And then focused mass spec measurements are performed in a two-dimensional array of all along this tissue section. And then in the end, you get an image that has, say, intensity data of each single of a molecule, as shown here. Yellow is a lot, blue is little. And you can, of course, generate with one experimental run these distribution data or intensity, relative intensity data for thousands of molecules. And although this prospect is of course, exciting, there are some limitations that, because as you can see here, that the lateral resolution is only now going from about 10 micrometers. So each of these pixels here is 10 micrometer square, and going down to cellular resolution is happening, but of course that's only evolving at the moment. And so this is one application from our own research of this lipidomic analysis. In our case, we investigated the cellular senescence of dermal fibroblasts. So we had replicative senescent fibroblasts, and then with one method, so with an HPLC-MS method, we identified, okay, that one type of lysophospholipid is strongly enriched in the senescent cells, and the cells going to replicative senescent growth arrest. And then our colleagues at the Technical University under Martina Machetti-Deschmann, they were able to localize then these ions within say skin sections. Again, you see yellow means a lot, blue means little. You see, you can here see that the epidermal compartment, the dermal compartment, and so that would be such an ideal workflow where we have then a kind of targeted localization of bioactive lipids which are generated in or which are detectable in cells that underwent a certain protocol. And in this same protocol, we also had we also identified also in other senescent cells, we identified lipid aldehyde species. So these are highly reactive molecules within a keto or an aldehyde group. And of course, we also wanted to localize these, and we wanted also to correlate their presence with, for example, molecular markers of cell senescence or cell damage in the tissue in situ, or with other modalities. And what we need for that is a kind of first of all, a definition of where is the playing field, and what do we correlate to. And so we wanted a kind of a segmentation of the epidermis into certain defined strata, and we want to see a label, for example, DNA damage at subcellular or cellular resolution, and we need some kind of prediction or real masks or boundaries of the cells to then be able to do analysis on that. And for that, one needs, of course, some kind of advanced image processing software. And our image-based multimodal correlation starts by here drawing annotations for an artificial intelligence algorithm, so that it learns where to recognize the epidermis in a section, and that was stained with DAPI, so with some nucleotide and with antibodies. And then in a second step, there is a nuclear segmentation performed by the software, and it predicts so-called cell masks. And from that, it also predicts then the strata. So depending on whether there are nuclei and what is the relative localization of the cell, it is assigned to either the basal layer or suprabasal layer. And then as soon as we have that say assignment, then we can add the data from other modalities to the experimentation. So in our case, we did two things. We did an enzymatic activity assay, so that's for some glucose metabolic activity, G6PD. We have an in situ assay that can be recorded together with the the fluorescence, or we try to correlate the data from the mass spec imaging, which is of course the most interesting for us. For the first approach that we, 2 years ago we showed that you can see whether the UVB irradiation of epidermis, or in that case it was an epidermal equivalent, whether it would affect the glucose metabolism in situ, so whether it would transfer so glucose through the pentose phosphate pathway. This would be this is a reported mechanism, a protective mechanism, because the PPP produces reducing equivalents which are needed for the antioxidant processes in the cell, and it also provides building blocks of for DNA, for nucleotide synthesis, which is needed again for DNA damage repair. And we correlated this then with a signal for DNA damage. So we did a gamma H2AX staining, and then the software identified the cell shapes and cell areas of those cells that undergo DNA damage repair. And using that, we could show that indeed, immediately after this irradiation, the PPP is switched on, and especially those cells that have DNA damage have a very strong signal of the pentose or G6PD activity and pentose phosphate pathway activation. And now we also applied the same, or a similar image-based workflow to correlate the data from the lipidomic analysis. So again, AI annotation, nuclear segmentation. And then, of course, it was very tedious. We have, because the resolution of the images is very, very different. It's on very different scales, and so we had to transform and warp the images to bring the MSI lipid intensity data in a form that it can be co-registered with the much higher resolving microscope data.
Florian,
then, yes.
Sorry to interrupt you. Just to see how many slides do you have?
This is the second before the end.
Okay. Just because we are running late on time.
Okay, sorry. And so using that, we could then really localize within the epidermis these lipid data, and we could find a position for each nucleus within the strata, a value for the intensity staining, in this case of, for example, loricrene, to look at differentiation. On the y-axis here you see loricrene intensity, here you see to which stratum it was assigned the lipid, and the color of the dot from black is low to red is high, shows us the intensity of the lipid signal that is in this detected in this area. And this can be then applied to thousands of signals, and we could therefore localize these lysophospholipid and the aldehydophospholipids that are associated with cell stress and cell damage. And so this is already my swan song picture. So there is, of course, challenges to this multimodal imaging approach. We chose an image-based approach. There are also other approaches, and people, for example, in the Comulis cost action are working on say defining uniform ways how to deal with the issues for registering say one modality, for example lipids, to another modality being fluorescence imagery. So this was just a short introduction into the topic. I'm a skin biologist. I'm not a mass spectrometry expert or a lipid chemist, but there is resources that give great training and information in these fields. Especially you can see them in epilipidnet, also in cost action, or lipid maps, and wiki pathways provide very nice say information if you are more interested in this field. And then with the thanks especially to Chris and Sam, Ionella, I want to, from my team, I want to thank them. if you are interested in redox biology, there is a meeting in Vienna that I organize pretty soon, and I want to thank the funding societies. And I want to thank you very much. I'm sorry if I was long, yeah, thanks.
Many thanks. Really, really many thanks. It was an amazing, very interesting presentation. You make us know, I mean, a new word, because people who are not inside lipidomics, I mean, it was very nice. And also the wiki pathways, so pathway for people, for the normal people who are not in lipidomics, are very interesting. So I don't know if we have time for questions. What do you think, Enikö?
I cannot see any questions now. Wait, maybe I can ask you just a short question, if that's fine. I was thinking a little bit about what you said, that the lipids, and I guess especially the lipids in the stratum corneum, are very much influenced by the environment, like UV and maybe even diet and other external factors. So when doing this lipidomic studies, how do you take this into account? For example, if you want to study lipidomics in atopic dermatitis or psoriasis, do you sample several times, for example, to or how do you take this into account?
Yeah, so if they are affected by environmental factors, and this manifests in chemical modifications, so this can be detected, for example, in LC-MS by oxidation, for example, of one's fatty acid. This chemical modification changes the properties. So it, for example, it changes the time that this lipid will need to elute from the column. And so there are not yet very advanced automated workflows that can then really specify, okay, this environmental modification, or this say inflammation-dependent modification of a certain lipid is kind of clearly identified on the molecule level. But this would be, say, this was the starting point for us, and then you have to go into details into more advanced say MS methodologies with positive and negative mode, and then you can really find out what is the structural change that was affected to the lipid by the environmental, for example, oxidative stress. But this is, so there is a possibility in say simple methods to see already big changes in for example oxidative modifications or epilipidomic modifications, and then the specification and the localization need then the other technologies I showed today here.
Thank you very much. I think really, as Anna said, this is a whole new word for many of us, and it's very exciting, very rapidly evolving.
Yeah.
So I think we don't have time for more questions, unfortunately, but this presentation will also be available later on YouTube. Thank you very much for your nice presentation. I have a few announcements to make after this, if you just stop sharing.
I stop, yeah.
Yes. Yeah, so, yeah, and before that, of course, happy birthday. This is a very special occasion. Yes. And this is the next episode of the ESDR Kitchen, which I would just like to make an announcement for. This will be on the 24th of May. Yes. And also another announcement for the state of the art in dermatology for Ukraine Solidarity Action of the ESDR. The day 2 of this online symposium will be on the 27th of April, and access is free, so you are very welcome to join, all of you. And last but not least, I hope to see many of you in Tokyo, Japan at the ISID 2023. Thank you again, Florian, for your nice presentation, and thank you all for listening today. Bye-bye.
Thanks a lot.
Bye.
Bye-bye. Thanks for having me. Bye.